The term fish is used to refer to a grade, that is, a non-monophyletic assemblage, of aquatic vertebrates with gills. The cartilaginous fishes, chondrichthyes, and bony fishes, osteichthyes, are both fishes in this broad sense. However, because tetrapods are nested within osteichthyes, a tuna is more closely related to a rabbit than it is to a shark. Generalizations across fishes should therefore be made only in light of this knowledge. This video gives an insight into the inner anatomy of ray-finned fishes, class Actinopterygii. The larvae of ray-finned fishes undergo metamorphosis. During this metamorphosis, the larval organs are resorbed or shed, and the adult organs are developed. A larva, which is still connected to a yolk sac, hatches from the fertilized eggs and subsequently quickly absorbs the yolk. Our first preparation object is the sterlet, Accipenser ruthinus. This is a species from the Sturgeon family, Accipenseridae. This species reaches lengths of 40 to 100 centimeters. It has a slender body with an inferior mouth. It has a dark gray to brown base color. The belly is yellowish to white with a red sheen. On the slightly upwardly curved snout, there are 4 barbels of equal length in a transverse row that extends to the front of the mouth. We will discuss the structure and function of these barbels later. This species has a total of 5 rows of bony plates covering the body. The gill cover does not completely cover the gills. The gill cleft always remains slightly open. As with all sturgeons, the caudal fin is asymmetrical, that is, heterocercal, with the upper part being longer than the lower one. The dorsal and anal fin are on the last quarter of the body. For the preparation, the sterlet is placed in the preparation tray, or alternatively can be held in one hand. With a fine pair of scissors, the left side of the body is opened behind the gills up to the anus. Make sure that the scissors are always angled outwards so as not to snag the internal organs. An upper incision is made above the lateral line, just below the back line. The abdominal wall is now cut deeper both behind the gills and in front of the anus, and pinned into the preparation bowl with needles. Once this has been done, we get a first look at the internal organs. We can see the ruffled pyloric ceca, which are part of the digestive system, extending from the intestine. The gonads near the kidneys stretch like a fine band from cranial to caudal. The bright red small intestine follows the gastric ceca and merges into the dark red large intestine. The swim bladder is an organ of the ray-finned fishes. It serves for buoyancy control. Fishes are able to adjust the volume of gas within the bladder and thereby compensate for specific gravity of their bodies and the surrounding water. It is derived from the intestinal tract and is homologous with lungs, but serves for hydrostasis, not respiration. It is easily recognizable as a whitish, transparent organ. In some ray-finned fishes, the swim bladder is still connected to the intestine, as we see in the sterlet. In others, however, this connection is absent. The kidney can be seen as a dark red stripe dorsal to the abdominal cavity, lying along the vertebral column. They can be either paired or unpaired and fused. In the sterlet, they are paired. The liver is beige in color and surrounds the front section of the intestine and the stomach. The individual organs can now be carefully removed from the abdomen by separating the mesenteries. At this point, the connection between the intestines and the swim bladder is clearly visible. Once the individual organs of the digestive system are exposed, the front part and the gills can be opened. To do this, the area around the pectoral fin is cut open with fine scissors. After the pectoral fin is removed, the gill cover can also be removed. Now the gill filaments, the heart, and the front part of the digestive system are exposed. With a freshly dead animal, the heart is sometimes still beating, as can be seen. This is not, however, an indication that the animal is still alive. The heart has its own excitation center, which evokes action potentials and leads to the brain-independent excitation of the heart muscle. To take a closer look, we will remove the needles so that the animal can be picked up. In this close-up, the arches of the gills and the beating heart are clearly visible. With a fine pair of tweezers, the fat bodies can be separated further from the digestive organs, and these can be laid out in their entire length. The elongated kidney and the gonads can be seen outside the abdominal cavity. The actinopterygians have developed ovarian and spermatic ducts in addition to the ureter. The reproductive organs are located to the side and above the intestine. Here you can see the ventral side of the head. The gill covers have been removed on both sides. The inferior mouth is surrounded by fleshy lips and can be extended far forwards. It is also equipped with a transverse row of 4 barbels, which are supported by cartilaginous rods and serve as tactile and chemosensory organs. The gill leaflets and the heart in situ can also be seen. Ray-finned fishes have a closed circulatory system, driven by a simple heart located close to the gills, which consists of an atrium and a ventricle. It pumps oxygen-poor venous blood directly into the gills, which are made up of numerous leaflets and capillaries. The gills absorb dissolved oxygen from the water over their large surface area and pass it on to the blood. In fish, the sinus venosus forms its own heart chamber, which is connected upstream of the atrium and in which the tissue that creates the excitation is located. In the ray-finned fishes, the conus arteriosus has largely receded. Instead, the bulbus arteriosus developed from the beginning of the aorta, a structure that contains many elastic fibers and smooth muscle cells. It creates a strong Windkessel effect to maintain blood flow. The gills are covered on the outside by the bony operculum. On the 4 gill arches, made up of bone and cartilaginous tissue, there are gill filaments on the edge in 2 offset rows. They contain a thin rod of cartilage and are covered by a multilayered epithelium. Gas exchange takes place over the thin gill lamellae, which stand on both sides of the gill filaments. In fish, the gills also have an excretory function. For example, they release ammonia to support the kidneys and can also absorb ions from the water. The second preparation object is the rainbow trout, Oncorhynchus mucus. This belongs to the salmon fishes, Salmonidae. The lateral line organ is a dermal organ involved in the external perception of fishes in water. In many fish, the sensory cells are recognizable as a line of pores on the lateral sides of the body, hence the name. The lateral line is able to perceive movement, vibration, and pressure changes in the water, and is based on epithelial cells called hair cells, which act as mechanoreceptors. A look in the trout's mouth reveals the fine teeth. Ray-finned fishes have 2 sets of jaws, the oral jaws that open and close the mouth, and the pharyngeal jaws, positioned at the back of the throat. Oral jaws are involved in food collection, whereas pharyngeal jaws are mostly involved in food processing and transport. Fish skulls are also highly kinetic, that is, they consist of many movable elements. Whereas in mammals, only the lower jaw is typically moved in food collection and processing, the upper jaw and parts of the cranium may also move during feeding in fishes. For the preparation, the rainbow trout is placed in the preparation bowl or taken in one hand. With the other hand, the abdomen can be opened using fine scissors. The lateral line organ can be seen as a line towards the dorsal side. We can cut above it in order to identify the muscles and bones. As soon as the left side of the body is opened, the fat, the delicate swim bladder, as well as the spleen and kidney become visible. The trout's swim bladder is very tender. The firm swim bladder of a carp serves as a comparison. The individual organs of the digestive system can now be separated from each other by severing the connecting mesenteries and removed from the abdomen. For a closer look at the gills in situ, they are fanned out with a probe. All organs are encased in a fine connective tissue rich in fat cells. The fatty tissue surrounds the digestive organs and, like the mesenteries, can also be carefully separated. In addition to the swim bladder, fat is also used for static buoyancy. The kidney runs as an unpaired, elongated organ between the spine and the abdomen. Recall the paired kidney of the sturdid we saw earlier. The lobes of the liver are also clearly visible now. The four-part heart is easily recognizable in situ. It is surrounded by its own pericardium. It consists of the sinus venosus, the atrium, the ventricle, and the bulbus arteriosus. The sinus venosus of the heart is noticeable because of its dark red color, and varies in size depending on how full of blood it is. The ventricle is more muscular, and the whitish, cone-shaped bulbus arteriosus sits in front of it and is red in color. Blood enters via the sinus venosus into the atrium, and thence into the ventricle. Contraction of the ventricle sends the blood into the conus arteriosus and then through the ventral aorta, from which it passes into 4 pairs of branchial arteries towards the gills. For a closer look at the heart under magnification, it can be dissected out. To do this, the area in front of the sinus venosus and behind the bulbus arteriosus is severed with fine scissors, and the heart removed. Finally, the 4 gill arches are viewed. The jaws of jawed vertebrates, that is, gnathostomes, are in fact derived from the anterior-most gill arches of early vertebrates. Our third preparation object is the common carp, Cyprinos carpio, a member of the carpfishes family Cyprinidae. The common carp is one of the most famous European fish species. It has long been a popular food fish, which is now kept in fish ponds and has also been introduced into numerous countries around the world, where it sometimes occurs as an invasive species. The native population, however, is now considered threatened. The anatomy of the carp is similar to the salmonid rainbow trout dissected before. Lifting the gill cover reveals the fleshy, dark red gills. A look in the mouth reveals the carp's tongue, which is actually a bony structure called the basihyal. There is a barbel on either side of the mouth. The dissection proceeds as for the trout. As soon as the abdominal cavity is opened, the fat-streaked gonads, the spleen, and the liver are particularly visible. The internal organs can only be disassembled, separated from one another, and removed from the animal's abdomen by carefully separating the mesenteries. The fat-permeated gonad can be removed from the abdominal cavity and separated. The gonads, in this case the testes, which fill the body cavity between the intestinal tract and the swim bladder as a milky white mass, narrow towards the rear and merge into the urinary duct, where they unite and open into the urinary bladder behind the intestine. Now the digestive organs and the powerful swim bladder are clearly visible. This can be carefully lifted and removed from the abdominal cavity. In carp, there is no connection between the swim bladder and the intestine. The swim bladder appears to be isolated in the abdomen, but is supplied by blood vessels. Our fourth preparation object is the zebrafish, Danio rario, another member of the Caprinidae. Zebrafish are native to the Ganges River basin in Pakistan, northern India, southern Nepal, and Bangladesh, where they live in slow-flowing or stagnant waters, such as rice fields. The zebrafish is used as a model organism in genetics and developmental physiology, and much that we know about human genetics, development, and disease can be attributed to studies on zebrafish. The zebrafish has an elongated body that is only slightly flattened on the sides and reaches a maximum length of 5 centimeters. The back of the fish is olive brown, the belly yellowish-white. The sides are traversed by yellowish-silver and blue to black iridescent longitudinal stripes that extend from the rear wall of the gill cover to the caudal fin. The anal fin and the gill cover are also striped or spotted in contrasting grey-blue and yellow. The chest and belly fins are colorless. The mouth is slightly superior. It is flanked by 2 pairs of barbels. As the zebrafish is so small, the preparation is carried out with fine scissors for micro-preparations, but in other respects the procedure is the same as we have seen for the other fishes. A light pressure with the fingers of the holding hand makes it easier to cut open. A ventral incision is made, which is then followed by a vertical incision behind the gills and dorsally in front of the anus. For further opening, the fish is placed in a dissecting dish. The previously opened abdominal cavity is now carefully stretched with pins or fine insect needles. If this takes place with the fish slightly tensioned from front to back using pins, the abdominal cavity can then be more easily stretched. As soon as the abdominal cavity is opened and a first view of the internal organs can be gained, the area behind the gills can be opened up with microscissors. The gill cover is then lifted and pinned in place with a fine needle, so that a clear view of the gills and the heart is possible. In order to distinguish the individual organs, they are carefully separated from each other with a fine needle. Gradually, the air-filled swim bladder, the dark kidney below the spine, the yellowish gonads, the ovary, and the digestive tract become visible. Our fifth and final preparation object is a flatfish, the common sole, Solea solea, a member of the Soleidae. The flatfishes form the order Pleuronectiformes, which stand out by their very unusual adult morphology, adapted to a benthic way of life on the sea floor. Adult flatfish are strongly flattened on the sides, and since both eyes are on the same side of the body, they are strongly asymmetrical. As larvae, flatfish still have a bilaterally symmetrical body shape and swim upright in open water. In the course of their metamorphosis, one eye wanders in front of the dorsal fin or through its base to the lateral upper side of the body. The skull bones as well as the outer nostrils shift in the same direction. Muscles and nerves, the mouth, the teeth, and the colored fins are also involved in this metamorphosis. In the left-eye flounders, family Bothidae, the right eye almost always moves to the left. In the case of plaice and sole, the left eye usually moves to the right. However, there are cases where either side can become the top. Meanwhile, the body flattens out. In most species, the swim bladder recedes. The common sole, Solea solea, lives close to the coast on sandy and soft soils, usually at depths of 10 to 60, more rarely up to 150 meters deep. Soleiids are right-eyed flatfish. The common sole has an oval, elongated body and a rounded head shape. The upper side is grey-brown to red-brown with indistinct dark spots. Their underside is light grey to white. The dorsal fin begins on the head in front of the eyes. For the preparation, the sole is taken in hand and cut open with microscissors on the left side of the body, that is, the side that does not have eyes. It is important not to cut too deeply so as not to injure the internal organs. After the gill cover has been carefully removed, the gills are exposed. The sole that has been prepared is now placed in a dissecting dish and pinned there with fine dissecting needles. With the help of the dissecting needles, the sole can now be gradually opened. Once the organs are exposed, we can note that the same structures as seen in the other fishes are present, but their positions are altered as a result of the dramatic rearrangement of the body in metamorphosis. Note especially that the anus is rather near the head in the flatfish. This is a result of the anterior migration of the anterior end of the anal fin, which begins just behind the anus and runs to the base of the tail. This concludes our overview of the anatomy of bony fishes in the infraorder Actinopterygii. We have seen that the basic internal anatomy of the bony fishes is rather consistent, but details differ across different families. When body form is rearranged, as in the sole, the basic elements are mostly retained, but simply rearranged. In the Actinopterygians, we can see many features that closely resemble the structures seen in tetrapods, much more so than in the cephalochordate lancelet. For comparison, we encourage you to compare what you have seen here with our videos on the anatomy of Branchiostoma and Tunicata, amphibians, reptiles, and mammals.